RNA carrying anticodon and then going through the polypeptides. So then we talk about translation. And I already said translation is a codon expressed as amino acids. So we need to go through some basic information. And we'll talk about later on when we come back, okay, when we have time. First of all, what is amino acids? You need to know. Some of you doesn't even know about that. Amino acid, what it looks like. The basic structure. CNH2, COOH, H, and R. That's a basic structure. The center C, chirocarbon requires. Well, there is an exception. Glycine is not chiral because the side chain is H. Okay, that's an exception. We just say the chiral C. What is NH2? Amino group. What is COOH? Carboxy group. And the COOH, we also could write like C carbonyl group and the hydroxy group like that. And this H is just a proton unit. The major difference to separate all the amino acids is this R side chain. That makes difference. So always remember, amino acids is major, only one place is difference, is this R side chain. Okay, how many amino acids then? Millions of, yes. How many amino acids related to our everyday life? Or for bacteria everyday life? There is 20 amino acids. Well, textbook right now tells you it's 21 amino acids. Okay, we'll talk about that later. Give the example. So 20 amino acids, let's just say that. Okay, so the job for translation is to using codon to express amino acids. How they use the codon to do is a 3-letter codon. 3 letters codon to express it. So this gives you a basic mass. 3 letters codon. I have 3 boxes. Each of them, I'm going to label them box number 1, box number 2, and box number 3. What are the codon from? Of course, coming from A, U, G, C. How many possibilities could it be the different gene base in this box 1? How many possibilities? It's 4, is that right? How many possibilities will be in this box 2? It couldn't be A, U, G, C, any of them, so it's 4. How many possibilities will be the last one? Same thing as a 4. So if I have a 3 letters codon, how many possibilities of this codons I will be generated? Multiply them together equals 64. So there is a question you learned about already. 20 amino acids expressed by 61 codon. You said there is a 64 possibility. How come? And the bacterial cell is always smart. They're not going to waste anything. Because we have 3 stop codons. What are they? UGA, UUA, and UAG, if I still remember. Let me see it. UAA, UAG, and UGA. That's 3 stop codon. Okay? Therefore, it becomes 61 codons to express 20 amino acids. Now the remaining another miracle question. I have only 20 amino acids. You have a 61 codons expressed to it. How come? This means there is some flexibility about it, which means every amino acid could be 2 to 4 codons to expressed or translated for one amino acid. But how they do that? For all the bacteria, amino acids, they are very strict for the first place and second place. And they give you a flexibility of the third box. This phenomenon, we call it some wobbling. Or we say code degeneracy. Or you can say some redundancy, degeneracy. So give you an example. GGA, GGC, GGG, GGU, all will be translated to glycine. And if you look at the code here, glycine, GGG, GGU, GGA, GGC, they're all glycine. And if you see AUU, AUC, AUA, it is all isoleucine. But something there's only one. What is that? It's a start codon, which is AUG. It's only one. Methionine is only one. Or methionine. And this is really nice. What happened? If there is during the DNA replication, somehow there are some mistakes happen. If luckily this mistake happened in this carbon number 3, the error will not cause the change of the amino acid, type of the amino acids. In other words, they still can survive. So this type of the mutation, we call it a silent mutation. It doesn't matter. It will still survive. However, if you have a codon which is mutated in the carbon number 1, carbon number 2, most likely you will generate a different amino acids. This is called a missense mutation. Is that right? So that's very nice. We don't want to talk about mutation too much because of time limited. So just want to tell you, the system is really nice. The code degeneracy, a little bit of redundancy, have a 61 codons to translate 20 amino acids, and they give flexible called wobbling at the number 3. It's really nice. Okay? So this is a wobble situation what happened. Okay, before we talk about the translation, we need to talk about the structure of tRNA, what it looks like. Then we can talk about the procedure. Okay, tRNA, it's a very nice structure. What the tRNA looks like. You draw like this. Like that. This is 1 carbon, this is 3 carbon. Okay, what are these? We know about this area carrying 3 codon, but anti-codon. So anti-codon arm. This area, variable arm we call it. This side is called T theta C arm. In this T theta C arm, you will see lots of the weird, we never heard about those gene bases. let's say PY, PU. So sometimes in the genetics, there's a tricky question. They give you a sequencing, AUGC, PY, PU. Then the second choice is AUGC, then ACGU, whatever. Which is a possibility of the sequence for tRNA? If you see any of them have those willed gene base, which is coming from the T-sequence arm, that could be transfer RNA. Okay, the last one, D arm. Okay, that's D arm. And this top connected with amino acid. Okay, that's a tRNA structure, what it looks like. Very important, before we talk about, we need to talk about something is how they initiate. So we talk about 20 amino acids. These 20 amino acids has to be carried on by tRNA to synthesis. Then they go to the ribosome to start to do the translation. So before they do that, they must have some reaction. This reaction is amino acid with tRNA and rely on ATP, and they will be hydrolyzed, and also rely on not hydrolyzed, combined, and need magnesium. They will generate adenine monophosphates and 2 phosphates, and then a very special structure. What is that structure looks like? That structure usually is hard to draw, but you have to understand what it looks like. Okay. This is AH, H, OH, H, OH, H, CH2, O, phosphate, phosphates. It's a ribose. Okay, so we call it nucleotides. What happens? This is here. Carbon number 1, carbon number 2, carbon number 3. This here going to be happening for something. Will be connected with something. Connected with what? Amino acid. That's carbon number 3 will be connected to amino acids. Okay, how about here? Carbon number 5. What is going to happen? This here will actually connect it to a tRNA. So this here, if you write a jar connected, I will be taking this off. Took this off. This goes here. Five prime, that's tr. This is the structure what it looks like. So what's the name for this? Amino acyl tRNA. And they need an enzyme to become this. Amino acyl tRNA synthesis. And this synthesis has to be unique and specific for 20 amino acids each. So that means we need 20 different amino acids, this amino acyl tRNA synthesis. But basically it looks like that. That's amino acyl tRNA. Okay, and during the development of the bacterial genome, they always started the first thing. It's very interesting. The first thing is always start from MET. And you learned in your other classes, the first transferring the amino acid is always MET, and it's a start code. But you need to know this is for human, animals, and in my class, this is for eukaryotes. How about for bacteria? Bacteria need to do a little bit of modification. We call it a formal methionine AEM. That is the first one. So what's the difference? Then we need to go back to know the structure of the amino acid. What the methionine looks like? CH3S, CH2, CH2. So you go here, you have a C, then you have a H. You go here, you have this guy, then you go TRNA, and then you go down, you have NH, then you go here, you have CH, then you have O. This is a formula group. So this is a structure, is always the first one to start to do the translation for a bacteria cell. It is different from what we talk about for the eukaryotes. Okay, now next, let's really talk about how they started to do the translation. Okay, we're going to use the whole blackboard to explain, so I take this off. So that's what we talk about here is the initiation, how we do it. The tRNA, amino cell tRNA synthesis, and also is the formal tRNA, methionine tRNA, what it looks like. So let's talk about really the translation, how they do. So initiation. Okay, maybe I do this side too. Okay, before we talk about translation, we need to talk about another knowledge. We have to review. So the question will for you, what is the location to do the translation? The ribosome. Ribosome is the location to do the translation. So what's the structure of the ribosome? This is we have to review. There is a 2-unit cell. Add up together 70S. 70 units, which means units to describe the speeding, the precipitation, not really the molecular weight, so this is not going to add up together. So 2 subunits on the top, 50S subunits, the bottom 30S subunits. Remember, not add up together. 30S subunits, you have a 16S RNA. It's very important. 50S subunits, you have a 23S RNA, also very important. And you also need to know, in the ribosome, there is 3 locations and 3 boxes important for the translation, which is we call EPA. And I said that this is not an environmental protection agency. It's just the acronym is accidentally are the same, but it's different. What is it? E means exit site. So at the end of the day, this free tRNA will release from there. Okay? What is P site? Transpeptidosite. Happen is transpeptation, which means there's a transpeptase enzyme for that. What is A site? Amino acid binding site. So those are the 3 locations you need to know. That is happened for the translocation. So let's talk about that. What are we going to do? First of all is initiation. once the initiation happened, we need initiation factor 1, 2, 3. So this 30th subunits is there. Originally, this 30th subunit is attached with initiator factor 3. And make sure it is separated with 50th subunits, not doing anything until something happens. So there is an initiator factor 1 gonna come in. They will turn this off and attach to it. So that's initiator factor 1, which means I'm gonna start ready to do the translation. Okay. How are they gonna do it? We need another initial factor 2. So initial factor 2 looks like this. I always say it's like a boat. This boat will carry on something, which is carry on a TRNA. Like that. And this TRNA is always carry on MET at the beginning. Okay, since this is a boat, when they go into some places, when they drive to some places, they need to carry on the money. What are the money they carry on? Is GTP. So at the beginning, what happened is this initial factor 2, like a boat, carry on a TRNA-MET or FMET, and then carry on the money, GTP, going to the ribosome, which is 30th subunits. And what happened? This GTP will hybridize, become GDP, and this initial factor 2 will be released. Okay, will be released to find another new FMET to start another cycle. So that's what they happened at the beginning. Okay, now let's talk about that. This 30th subunit is right here. And there is an mRNA going to come in. But you need to know, it does not mean every single mRNA will go through translation. The mRNA, to start to do the translation, they must have some special sequence to start it. So the mRNA requires you to have AGG. This is a very special sequence. We call shine the Garno sequence. Acronym just say SD sequence. This Shine-Garno sequence must be recognized by 16S RNA in the 30S RNA subunits. Okay? This has to be recognized by 60S RNA. Then they can start to do translation. So if mRNA has AGG when they go there, what happens? This 50S subunits will be covered. At the same time, originally this elongation factor 1 is attached to the side of the 30S subunits. They will be released. So this elongation factor 1 will release. So once this guy has been packaged, 30th subunits at the bottom, 50th subunits at the top, and at the middle there's an mRNA, and you have AGG, then they start to do the translation, which means the second step is elongation. Now what's going to happen? So we're going to draw a couple of things right here. Okay, let's draw on the top. I have this sub-units right there. 3 position E, P, A. Okay. Of course, this is a large one, okay? So this is 30s, this is 50s. And there is some TRNA going to carry on, MET going inside. Let's just say MET. So they going through here. So what happened? Go here, go here. So this is MET. Let's say the first one. Go to the P position. Okay, then I have another very hard-working TRNA. They're going to carry on amino acid coming. So I'm going to draw right here. This is the second one. That's carry on, let's say, amino acid number 1. Okay? So the second step, they're going to move. How are they going to start to move? We're going to have to draw again. Okay, so I draw line right here. Big one, then E, P, A. So how they move? They will be moved this direction. And the synthesize is always 5' to 3'. Same thing as replication and transcription. So they move this direction. How they move? How they move it? This guy will actually attach to you. Okay. So what it looks like? This original one already been carrying MET will move to this side. And then they will later on will release. That's why we call it the action side. So on the P side, what is going to happen? On the P side, is actually happens is the mRNA will be AA1 and MET at the top. And this is 5', this is 3', remember? And then you have a new one going to come in. A new one will be right here. So carry on AA2.